Hello, and welcome to the Short Read Workshop Day 1 video for designing a sequencing experiment. I've talked in previous videos about how to prepare your libraries and how the sequencing works, and now I want to go a little bit more in depth into what these factors actually mean for designing your own experiment, specifically the relationship between read depth and what you're expecting to get out of your experiment, and how that may interact with these other factors as well. So here I'm going to give some examples of different types of sequencing experiments you might want to run, as well as best practices, bare minimum read depth that you want in mammals in order to get something that you can analyze out at the end. So for RNA-seq, if you're doing basic differential expression analysis over the general transcriptome, then you want about 20 million reads per sample at bare minimum. This changes if you want to look at something slightly different within your RNA-seq, such as if you want to focus on low abundance transcripts that may not be well represented in your pool of RNA, then you're going to want more reads per sample in order to try to extract more of those low abundance transcript reads. Similarly, if you want to do isoform analysis in your RNA-seq, then you need a lot more reads because you're going to get a lot fewer examples of reads across splice junctions, and you're going to need more sampling in order to extract that information. So at absolute minimum, you want about 50 million reads for these types of experiments. So for whole genome sequencing, you generally want at least 15 times genome coverage in order to estimate well any variance at any given position. And coverage is another way of estimating how your depth and your genome actually correlate. So for example, 15 times coverage of the human genome, which is 3.2 billion bases, if you have 75 base reads, then that corresponds to 640 million reads per sample. This changes depending on how long your reads are or if you're in a different organism, which is why whole genome uses a genome coverage calculation. So if you're simply looking for homozygous variants, 15x genome coverage is a good minimum number. If you're looking for heterozygous SNPs or variants or indels, then you're going to want much higher coverage in order to well sample any of those types of variants. If you're doing a ChIP-seq experiment, best practices are at least 12 million reads per sample. But again, if you're looking for broader peaks, then you're going to need more reads in order to well estimate those types of peaks. So you're going to want at least 30 million reads per sample. And if you're doing Hi-C, which is a 3D chromatin configuration type of sequencing, then that requires a lot of depth, at least 100 million reads per sample. So hopefully you can see, based on all of these different types of applications, that they're all going to need different read depth in order to really get the data that you want out at the end. So then this comes into the experimental design setup when you think about your actual sequencing machine. An Illumina Hi-seq generally outputs about 1.5 billion reads per sequencing experiment. An Illumina NextSeq outputs approximately 400 million reads, and an Illumina MiSeq outputs approximately 25 million reads. So the instrument that you're using, as well as the money that you're willing to put onto that instrument, gives you the number of reads that you can get out, and then you divide those number of reads by how many samples, conditions, replicates that you need, and that's how you get into all of these factors. You need to make sure that you have enough read depth to do the downstream analysis that you really want to do with your sequencing experiment.